Thank you, Michela, and I want to thank the organizers for the kind invitation. So today I'll try to give you an overview of what's been done in the field of HIV vaccines based on mRNA. And as all of you are aware of, creating a protective vaccine for HIV has been an extraordinarily difficult and challenging task. And this is due to the fact that HIV is really a master of trickery, disguise, and immune escape, which we can, I hope this is working, well, maybe not. To a series of devices and tricks. It has to protect the most important neutralization epitopes, especially the very prominent glycan shield, covers almost all the surface of the envelope. This conformational camouflage, the ability to only transiently expose certain epitopes, and steric occlusion that protects the most important ones. Antigenic variability, of course, and other tricks. So altogether, this has been a real nightmare. And OK, this goes back, so this should go forward. OK. And this really begs the question, is a protective HIV vaccine feasible at all? And the answer is actually yes, although it is extremely challenging. So how can I be so reassuring in saying that this is really feasible? Because experience has accumulated over the past few years that we can actually isolate broadly neutralizing antibodies, or BNAbs, which are directed against different sites of vulnerability on the envelope surface from infected subjects. This means that the immune system of humans can develop this kind of antibodies. And if you take one of these antibodies, even better a combination, and passively infuse macaques, you can protect 100% of the animals from infection with the virus that bears the HIV-1 envelope. So 2 + 2 is 4. It's really a simple equation. The main challenge is how can we develop such broadly neutralizing antibodies? And of course, I'm often asked this question, as Tony has discussed, can mRNA overcome by itself the major obstacles on the way to a HIV vaccine? And the answer is clearly no. But it can be of great help. And the reason for this is illustrated in this slide that was kindly provided by Moderna, because mRNA allows for something that is extremely important, especially for HIV. One, the possibility of expressing the envelope trimer in its membrane-anchored form, which is exactly what we find on the surface of the virus. And two, which is really important for HIV, this protein will bear the native glycosylation, because your body becomes the factory of the vaccine, and it will glycosylate complex glycoproteins exactly as they should be. This is important because glycans are integral part of some of the most important neutralization epitopes. So this is in addition of the general advantages of the mRNA platform. You heard a lot about safety, versatility, rapidity of development, efficiency, lack of immunogenicity, low cost, and you name it. So it's really an excellent platform. However, it remains the messenger and not the message. So it is a platform. It cannot solve our problems. Being so interesting, a number of studies have already been performed either in preclinical non-human primate models or in human clinical trials using mRNA in the HIV vaccine development. I will focus on some of these studies, but let me step back for a second and tell you about the main strategies that are being pursued for an HIV vaccine, regardless of the platform, mRNA or else. There are germline targeting vaccine, there are lineage-based, epitope-focused, structure-based, and of course combination strategies. And each group has championed some of these strategies. But I want to show you maybe a more philosophical slide, because I see in the field really two different approaches. One is what I call analytical approaches, which try to optimize each individual step of vaccine development independently, and then test for correlate markers of protection after addressing one specific step. The other is what I call the holistic approach, which attempts to optimize individual element in the context of a full vaccine platform. And this is the only approach that allows to test for real-life protection and perform challenge studies. An example, which is very successful, of the analytical approach is what Bill Schiff and colleagues are pursuing at Scripps. They are trying to optimize and perfect the very early step of vaccine development for HIV, which is the recruitment of germline antibody precursors. In this case, they focused on VRC01, which is a CD4 binding site-directed antibody. They developed and engineered a molecule called EOD-GT8 and multimerized it, which is extremely effective in recruiting these precursors in knock-in mice and in humans. They showed in this clinical trial that as a protein, EOD works very well and expands these precursors. And they have recently started an mRNA-based vaccine using exactly the same molecule. So this is one of those situations where you can directly compare the two results. And the study is ongoing. They actually have two, IAVG-02 and G03, which are ongoing. But this, again, is really just focusing on the one step, the very early recruitment and expansion of the precursors. And it's a long way to the finish line, of course. A similar approach, which is also very interesting, is what was developed by Bart Haynes and colleagues, and also Norbert participated in the study with a different target in mind, an antibody against the V3 glycan supersite, the H270, which in knock-in mice vaccinated with mRNA expressing an envelope that targets this precursor, gave some very promising results. And I understand that a clinical trial is underway, just started using exactly the same strategy. And then Bill Schiff and colleagues are also testing in a separate trial a full-length trimer, not just the EOD outer domain molecule, in three different forms. One is the soluble trimer, the other is membrane-bound, which may be more effective, and then a CD4 binding site knockout. But again, this is one step, and it's really using one form of trimer that is not a germline engager. So it's still an analytical approach, if you want. On the other side, holistic approaches are what we have embarked in a few years ago when we started to discuss with Moderna the possibility of developing an HIV vaccine. This was back in 2016. And we developed what we call a multiclade VLP-forming MGAG mRNA vaccine platform. What are the key features? One is we use exclusively membrane-anchored full-length envelope, and this is because it is increasingly emerging as the best form to elicit broadly neutralizing antibodies, certainly better than soluble monomeric GP120, but also better than the soluble trimers like the SOSIP, which indeed elicit large amounts of off-target antibodies. Second, by using mRNA, we can express the bona fide native protein envelope trimer in vivo, especially with the native glycosylation, as I told you before. And then we tweak the mRNA technology to produce virus-like particles, which are believed by many to be better immunogens than subunit vaccines. And how we achieve that, we asked Moderna to co-formulate in the same LMP both envelope and GAG. And GAG itself is sufficient to promote the formation of VLPs, as the one represented on the right side, which in this case are actually rich of spikes, of envelope spikes on their surface. Then we prime strictly with germline engaging envelope, which really start the process of affinity maturation of the BNAbs. And then we follow up with an intensive boosting at high dose with selected heterologous envelopes from different clades. This is because we want to focus on what is common between all these different envelopes from different clades. They are all tier 2, and selectively expand antibodies against the shared epitopes, which are, by definition, the BNAb epitopes. So in December 2021, we published the results of a first limited scale vaccine trial in macaques with this platform. One first good news is that the reactogenicity of this vaccine was very, very low, despite the high dose of mRNA that we used, especially per kilogram. We barely saw any significant side effect, and certainly not systemic side effects. And let's go straight to the most important data. We did observe at some point, only after the third heterologous boost, which is 6 vaccinations down the road, the appearance of broadly neutralizing antibodies. Unfortunately, they remained at relatively low titers, below 1 to 100, but it was true breadth. These are 11 of the 12 Montefiore small global panel envelopes from all over the world, different clades, and it's really remarkable that we almost all the animals could neutralize all of them except for one. This was associated with a very robust and polyfunctional CD4 response against envelope, including T follicular helper cells. On the bottom right are illustrated the TFH that were very robust, and we know mRNA vaccines are particularly good at that. And then we went all the way through, and we actually did challenge, and we used a virus, Shiv88, which poses the bar really, really high, because Shiv88 is a virus that the vaccine people stay away from because it's so difficult to neutralize. But on the other side, it's a real-life envelope. If we use an easy target, you may like to win easy, but then it probably doesn't mean much. This is a real challenge virus. And yet, we did observe a significant protection. Number of animals was low. The protection was not absolute. Only 2 animals did not get infected at all. This is a challenge with 13 weekly low-dose challenges, mucosally, rectally, and the controls were all infected after 42 days, but there was a significant delay and a calculated per exposure risk reduction of 79% overall. So this was successful. We were happy, but we knew it was just the beginning. So we need to further improve the platform, which is promising, but certainly far from perfect. And what we focused on in the last couple of years was how to make better VLPs, because the VLPs we were making were mostly immature without GAG processing. The HIV and the VLPs remain immature. But if you process GAG using the viral protease, you can obtain mature VLPs, which seem to be better immunogens. We actually have proof in mouse models, so I don't have time to show you. And this may be due to the fact that the envelope is free to move on the surface and may cluster. And clustering is important for B-cell stimulation. So what we did, we added one element. Instead of using the protease, which may be too toxic, we use GAG-POL, the physiological mechanism used by HIV, and now we have a triple formulation, GAG, ENV, and GAG-POL. And these formulation in knock-in mice, experiments done by the Stamatatos group, were extremely effective. On the far left, it's our formulation. The same envelope was tested in nanoparticles. They are used in a clinical trial only after repeated heterologous boosts. So this tells you how difficult it is to induce heterologous NUTs, but we did observe them again, but again at low titers. We did observe again very robust Th1 memory CD4 responses and TFH responses, and also CD8 responses, much lower than the CD4, but they were clearly detectable. And again, we observed protection, but here I just pulled arm 2 and 3 because they both have the GAG-POL, the protease. The protection was only observed in the presence of GAG-POL and not with ENV and GAG alone in this trial. There are many reasons I could discuss, but this really was important for us because it confirmed that the addition of the protease is beneficial. And this time, probably because of the high number of animals, 30 altogether, we did observe a correlation with neutralization of tier 2, including the challenge virus 88. And also, we were able to study the mucosal IgA and IgG, and they both seem to correlate with protection. This is not neutralization. It's too little material. It's just binding to the trimer, but it is clearly correlated. So that's very promising. That's why we are interested in boosting even further the mucosal immunity. And based on all these results, we have now started to design. We are in an advanced phase of designing and preparing for approval a phase 1 clinical trial that will be designated HVTN 310. And that will include, for the first time in an experimental clinical trial, 5 sequential mRNA immunizations. Why so many? Because it's HIV. And unfortunately, we cannot compromise for less. I know FDA will not be happy, and we are ready. We are rolling our sleeves for the discussion. This will be done with the triple formulation and with the most successful envelope roster that we used in macaques. So in summary, inclusion of viral protease is beneficial. The VLP-forming vaccine is extremely efficient in knock-in mice. And in macaques, it induced heterologous tier 2 neutralization, albeit the titers were low, and we certainly need and can hopefully improve that. Robust envelope-specific T-cell responses, TFH responses, CD8 responses, although low, and especially protection, which at the end of the day is the only thing that really counts, regardless of the correlates, from heterologous tier 2. Protection was this time correlated with neutralizing antibodies. So I'm not saying that it's only neutralizing antibodies. They probably are part of the equation, but certainly they seem to be important, and especially mucosal immunity is important, and the phase 1 clinical trial is scheduled to start in early 2024, FDA allowing. This is the long list of precious collaborators, Peng Zhang in my lab, and many others, Moderna, of course, Duke in many labs, the animal people, the Gates Foundation supported the second clinical trial, and especially I call them my custodian angels, the HVTN 310 clinical team that is helping to prepare this clinical trial. Thank you very much.